Cell signaling in epilepsy. There are three types of cell-to-cell communication: endocrine signaling, paracrine signaling, and synaptic signaling.
Endocrine signaling, a signal is sent throughout the whole body by secreting hormones into the bloodstream of animals or through sap in plants. Endocrine cells that produce the hormones secrete it into the bloodstream, making it available to bind to other cells. In paracrine signaling molecules are released from paracrine cells. These then diffuse locally through the extracellular fluid, targeting nearby cells. During synaptic signaling, an electrical signal moves along a nerve cell, triggering the secretion of neurotransmitter molecules carrying the signal. The molecules then diffuse across the synapse, triggering a response in the target cell.
Ligands and protein kinases. Protein kinases are enzymes that modify other proteins by chemically adding phosphate groups to them, thus changing enzyme activity. A ligand is a molecule that binds to the receptor, and during signal transduction, this causes a change in the protein, releasing an alpha subunit. Local and long distance signaling. Alright, direct contact is a form of local signaling which includes plasmosmata in plant cells and gap junctions in animal cells. Gap junctions connect the cytoplasm of two cells to allow the ions, molecules, and electrical impulses to travel across a regulated gate between the cell, directly affecting neighboring cells. Long-distance signaling in animals is possible through the secretion of hormones that seek target cells throughout the body, which also directly affect neighboring cells. Once these hormones reach their target cells, they bind to receptors on or within the target to make a change in that cell.
Ligand-gated ion channels. The receptors responsible for epilepsy are the gated ion channels, specifically the sodium channels, which are responsible for the majority of electrical impulses to the brain. The ions are funneled through these channels to enter the cell, as they are otherwise impermeable to the membrane. This movement is usually facilitated by ligands bonding to a receptor, which open the gate to the cell. This allows signals to be released into the body, permitting the human body to function normally. To close the channel, the ligand dissociates from the receptor.
The first step on a signal transduction pathway is reception. During reception, a chemical signal or ligand binds to a receptor on or inside its target cell. In this example, our ligand is binding to a G protein receptor. The next step is transduction, where binding causes changes among different molecules in a signal transduction pathway. In our example, when the ligand docks with the G protein, it causes a release of an alpha subunit attached to the bottom of the G protein. The G protein and an active enzyme are altered upon receipt of the signal. Finally is a response, where the transduced signal causes a specific cellular effect. In our model, the release of an alpha subunit causes the protein adenylyl cyclase to move down the membrane. Before this happens, adenylyl cyclase is inactive. Once activated, it attaches to an inactive enzyme, causing it to activate. It then converts ATP into cyclic AMP. Cyclic AMP then acts as a messenger to spread throughout the cell. These messengers then target protein kinases, and cyclic AMP binds to the regular side of them. This causes a release in the catalysts, creating a cascade of energy, or a protein kinase cascade. The catalysts pick up ATP, becoming phosphorylated. They are now able to act on enzymes within the cell. This protein kinase cascade changes the original protein that had caused the signaling. Epilepsy. Epilepsy appears not as a genetic trait, but as a mutation, making it difficult to predict inheritance in a family. One symptom of epilepsy is seizures, which can come in many different forms. Epilepsy is also the fourth most common neurological disorder, and it's characterized by the unpredictable seizures and can instigate other health issues. Epilepsy was first written about by Hippocrates around 400 BC. What causes epilepsy? One possible cause of epilepsy is the damaged ion channels in the brain. These channels, when functioning normally, produce electrical charges at a steady rate, sending information to all of the body cells. When it malfunctions, however, a chemical imbalance is created, causing nerves to misfire and leading to erratic chemical impulses and subsequently seizures. The malfunctions are caused by mutations in the human genetic code modifying the ions. The ion channels send signals to the brain, and the channels that, when mutated, have been linked to epilepsy are the sodium channels. This is because sodium is a very charged molecule and therefore is mainly responsible for electric signaling. Symptoms of epilepsy. The symptoms of epilepsy are staring spells, temporary confusion, and seizures. Someone is said to have epilepsy if they have 2 or more seizures separated by at least 24 hours. A seizure is the disruption of electrical communication between neurons. Seizures are unpredictable, episodic, brief, and stereotypic. They can often be manifested as odd behaviors such as repeating words, being unresponsive to people, speaking gibberish, and screaming. The end.
Goodbye.